Christina Rondinone. Christina is from MedImmune. Please join me in welcoming Christina.
Thank you. Thank you. Thank you so much. And I want first to thank the organizers for inviting me to the Peptide Symposium. And also, I want to tell you that it's great to be here because I have so many friends from the past and the present. And as you know, before joining MedImmune, I was in Roche, and I have the pleasure to work with Walid in many of these peptides. And actually, I have to thank, my ex-collaborators for the knowledge that I got and how I entered into this area. So I want to talk today about peptides and new therapies for metabolic diseases because I believe that for all of you that are peptide chemists, there is a huge opportunity here to really help us find and develop the best drug for especially cardiovascular and metabolic. And I will tell you a little bit more why. Why biologics? In this area, this is a huge unmet medical need. We need new, safe drugs, and we really need disease modifiers. And until now, what we were trying to do mainly is to treat the disease, but not go into the deep understanding of the pathophysiology and try to actually stop or reverse the disease. And I truly believe that in the case of biologics, peptides have a huge opportunity to do that. And then, why peptides? Because of the hot new discoveries in this area in the last, I will say, 2-3 years. Every single paper that comes up that is exciting is talking about a peptide or a good peptide or a bad peptide or a good protein or a bad protein that actually you can target with different analogs or with different antibodies. So I believe that this is extremely exciting. And I personally believe that the future of this area is in biologics. So this is great for you because everyone will have a job for a long, long time. And I will not say the same for actually medicinal chemistry. I think that it's getting harder and harder to get great targets in this area for small molecule targeting. So why we are here, you see the global medical need, and this is really an epidemic. If you see about the map of US and diabetes, you can see here that actually it's getting bluer and bluer, I will say. With more than 15%, 20% of the population with diabetes, especially in the southeast states, and this is growing. Yesterday actually I was driving and I heard in the radio every 4 people in the US are overweight already, and this will carry obesity, diabetes, and cardiovascular disease. So this is a huge problem. And this is not only from US, you can see the map of around the world. India, China, with those huge populations also, this is an epidemic, and the population is growing with the prevalence of diabetes. And as you can see here, more than 300 million are expected to have diabetes in a few years. So, huge problem, not only because of the epidemic and the patients, but also because of the societies and how much you will spend to treat these patients. So, as you know, type 2 diabetes is associated with many complications, and actually Andrew and Wolfgang talked a little bit about this before. And mainly, you have here diabetes retinopathy, diabetes nephropathy, but also we cannot forget about cardiovascular disease. And when you think about this, a huge proportion of the patients with type 2 diabetes will die because of cardiovascular event. So this is a huge problem too. And I want to show you this because many of you that are not in the field will think about why we need new drugs. And I hear that all the time. And if you think about how many patients achieve the treatment goals, are very few. And here you have HbA1c, you have blood pressure, and you have total cholesterol. But when you put all these things together, you have like more than 90% of these patients that actually will not achieve the goals. So it's telling us that the drugs that are over there are not good enough to try to solve the problem. And when we talk about diabetes per se, what I can show you here is that when diabetes is diagnosed, we already have, or the patients already have, half of the beta cell function. And then treatment with different agents like metformin as a monotherapy, and then you continue adding new drugs, and you finish with insulin, there is a gradual deterioration of beta cell function. So actually today, and we will see what happened with GLP-1s because we don't have enough data yet, but today we can say that actually we don't have any drug that will stop or will reverse the disease. And I personally believe that biologics and combination of biologics can do this. So this is what I told you before. Current therapies, especially insulin and segregagogues, produce hypoglycemia. Current therapies don't have long-term glycemic control, so then you need to add more and more and more agents on top of. There is actually some therapies are associated with weight gain. And then there is an increase of cardiovascular risk, and few therapies actually lead with an effect in obesity and body weight reduction. And on top of that, every single therapy have some safety issues, and actually we need to try to have safer drugs for both obesity and diabetes. So what we want to really have is drugs that actually will go beyond glucose lowering, that will control not only HbA1c, but will reduce weight, will have preservation of beta cell function, will have an effect in dyslipidemia and lipotoxicity, and also will have cardiovascular protection, and this is key. Because as I told you before, with type 2 diabetes, most of our patients die of cardiovascular disease. And scientific rationale, why biologics? And because, many proteins, peptides are secreted from many organs. And here is actually, I'm showing here the liver, where you secrete a number of different proteins, peptides that are very important not only for metabolic, but for cardiovascular and dyslipidemia. In the case of PCSK9 is one of them that is very, very hot. And as you know, everyone is developing like different antibodies against this target to reduce LDL in dyslipidemic patients. But then we have the fat and we have the gut. And the fat is one of the most important organs that actually produce and secrete a lot of, I will say, adipokines, good and bad. And the question is there any link between obesity and insulin resistance? And yes, almost all people that are obese are insulin resistant. And when you reduce weight, you improve insulin sensitivity. And adipocytes are really an endocrine organ secreting a lot of these adipose cytokines, having very profound effects in insulin resistance, dyslipidemia, inflammation, hypertension, and actually coagulability. And if you think about these are like a number of different fat-secreted factors. Most of them are bad, that are in red. And actually, maybe we can develop some antibodies that can contrarrest this effect. And all these factors are mainly involving inflammation and insulin resistance. On top of that, what we know today that is extremely exciting and has been like the research in the last couple of years is the actually moving the white fat to more a brownish fat and the brown fat. And actually it seems that many factors are involved in that process. and actually producing a more like energy expenditure adipocytes where actually you can reduce weight, increasing energy expenditure. And as I told you before, this visceral fat can induce or produce these factors that also can affect some kinases. And this is good news for the small molecule people that actually can target these bad kinases and can contrarrest the increase, for example, and I am giving you the example of the liver, but actually this is in many organs, in steatosis and mainly hepatic insulin resistance. So what about the gut, and why the gut? And is the gut the cause of diabetes? And this is very, very exciting. I believe that everyone in the diabetes field forgot the gut for many, many years. And they were talking about the liver, the pancreas, whatever, but you have to remember also that the pancreas comes from the gut. Actually, during development, the pancreas is formed by some cells in the gut. And then what you can say is that actually the gastrointestinal tract per se is one of the largest endocrine organs that we have. It secretes tons of different proteins and peptides, and has been like a revolution in the last years because of the bariatric surgery effect. And why the gastric bypass surgery? Because as you know, many of these patients, depend on the surgery, but many of the patients, like 90%, 98%, depends on which is the surgery done, actually are cured, and are cured after 2 days or 3 days after surgery. So these people are still fat, so they don't have any reduced body weight, but they go home and they don't take any other medication. So the question is, what is going on? What is going on so fast that actually you are cured? And that cure actually is maintained for years. Actually, now there is data of 11 years, 12 years after bariatric surgery that these guys don't take any other medication. And the question is there any change in secreted peptides? And here, there are some of the known secreted peptides that can be changed, and you see here our GLP-1 that we talked actually earlier today about this great peptide. But there are so many other ones that maybe are involved on this. And why? Because remember, from the 900s until now, there have been like a discovery of new peptides every single year. And actually, for most of these peptides, nothing is known about the function. So just now we are beginning to dig on the biology and what are these peptides doing in the organism and actually in the pathophysiology of obesity and diabetes. And to make it worse, many of these peptides actually are secreted by the enteroendocrine cells. And actually, if you look at the enteroendocrine cells, this is one enteroendocrine cells. These cells actually are like 15 different types of these cells, and actually are only about the 1% of the cells in the intestinal epithelium. So imagine how important are these cells and how actually few they are. And they are producing so good effects, secreting some of these factors. And then we have all these different cells in the intestine that we don't know still which are the factors that are secreted by these cells and can have an effect not only in bariatric surgery but in obesity and diabetes. So to show you, what I said before a little bit about the effect of these surgical procedures, you can see here that actually there is almost a resolution of type 2 diabetes after bariatric surgery, but it's not only type 2 diabetes, it's also hypertension, it's dyslipidemia, and actually cardiovascular. Actually, just now there are some reports showing like a reduction in cardiovascular events after bariatric surgery that actually are very, very exciting. So the facts about bariatric surgery, yes, today is the most effective method mainly to reduce body weight, although there are patients that are resistant, and will be very interesting to know why they are resistant and if there is any effect in these peptides. The weight loss maintenance is at least for 15 years. And then bariatric surgery mainly stops the disease and reverses diabetes. Because actually these guys, don't take any medication for a long time after. There is no drug that can mimic that today. And as I told you before, almost 70–80% of these patients are cured. But not everything is good about bariatric surgery. Bariatric surgery is indicated by those patients that are morbidly obese, just now, and diabetics. Depends on the countries. Actually, there are countries that just now they are having surgery in type 2 diabetics that have a BMI less than 27. So these are lean diabetics. But actually, because of all the other comorbidities that they have, they go to surgery because there is no drug that can stop them from dying because of diabetes nephropathy or cardiovascular disease. So just now, actually, there are countries that they perform diabetes surgery in these patients. There is some morbidity and mortality associated with the surgery, so we don't have to forget about that. It's not reversible and have sequelae, and they have effects in the bones, vitamin absorption. And then there is a risk, as I mentioned here, of insulinemic hypoglycemia with nesidoblastosis. So actually, and this is like an overgrowth of the pancreas that actually there are some patients that after 1.5 years of surgery, they have all these hypoglycemic episodes, and actually they found out that they have like a super growth, I will say, of the pancreas, and they have to have pancreatectomy done every 6 months or whatever. So there are maybe like super responders of this surgery. So the question is any opportunity for biologics here? Try to mimic what we have, or at least a little bit of what happened with gastric bypass. And I've got peptides involved in this bariatric surgery. And actually, Andrew mentioned before, all these different peptides that are secreted by the gut, and maybe, not only one, but the combination of these different peptides can mimic what is going on in bariatric surgery. And some data is there, of course, and one of the data show us that mainly, GLP-1 and PYY are increased after a bariatric surgery, and this is actually measuring this, these peptides even next day after bariatric surgery. They see already like a very interesting increase in these peptides. And then if you compare different type of surgeries, these surgeries that are more restrictive and these ones that are malabsorptive surgeries, you can see that actually the ones that are most effective, that are these ones, to cure diabetes, they have like a very interesting increase in GLP-1, in PYY, and also in oxytomodulin. It's very confusing about ghrelin, what is going on, but these three are always up. And to remind you, oxytomodulin actually binds to the GLP-1 receptor, but also to the glucagon receptor. And this is a very interesting molecule to learn from. So in summary of this part, I mentioned you the gastrointestinal tract is one of the largest endocrine organs. Gastric bypass is the only way to mainly cure diabetes today. And the resolution of type 2 diabetes is attributed to changes made in this secretory pattern after surgery. And we have mainly PYY, GLP-1, and oxytocin upregulated in these patients. So how can we target actually peptides and GPCRs? And all this story, I believe, begins with a GLP-1. I will not talk about what all the great things that GLP-1 is doing, because you know, but what I want to tell you is that I believe that has been like a revolution over the years about this, because when actually GLP-1, all the GLP-1 started, who believed at that time that actually a drug that will be injectable twice a day, was causing vomiting and nausea, will be a drug for diabetes, that is a chronic disease. And everyone was very skeptical. Only amylin, I believe, that was the one that developed this at the beginning, because everyone was so acceptable. But why is a good drug? And mainly because it's effective, very effective, have great effects, and actually is not inducing hypoglycemia. And this is a very important point. You know, there was I don't know if any one of you went last year to the ADA and was in that symposium that they debate what is first, insulin or GLP-1s. And this was like 5,000 physicians all over the world. 99% voted GLP-1s. And mainly why? Because of the hypoglycemic episodes that actually can lead to cardiovascular events. And for that, you know, GLP-1 can be like a great drug, and it is a great drug. And for that now, I'm glad that we have it in our portfolio from, you know, the amylin colleagues. So, and, you know, we cannot forget the medicinal chemistry guys. And have been many attempts with small molecules over the years. And you see here, you know, this compound, you know, BOC5 that binds to the orthosteric site, and then CPD1 binding to allosteric site. This is from a Chinese group, Chen et al., and this is actually from Knudsen. And actually, these compounds, what is known is that these actually will compete with GLP-1 per se to the binding site, and actually this compound binds really to the allosteric site and actually is complementary to GLP-1. But these compounds, actually what we know is that yes, you can have beautiful effect in vitro and maybe acute effects, but then you know, the PK are not very good. You know, they always have some problems to move forward. The same actually for these Lilly allosteric compounds that they show very nicely their effect in vitro and in acute animal models, but it's questionable how they look in chronic experimentation. So actually, you know, I want to show you a little bit, moving back to the peptides, and the question is how we can be better than GLP-1. And we have all the GLP-1s, as you know, Andrew and Wolfgang talk about, you know, the ones, the twice a day, once a week, once a month. Maybe it's coming the once a year in 10 years or whatever, but how we can be better. And one of the answers is maybe we can combine all these peptides together that I showed you before and have dual, triple peptides in one entity. And I want to show you, the proglucagon gene, mRNA, and protein. And what we know that there is tissue-specific post-translational processing in the pancreas and the intestine, producing these different peptides, including glucagon, GLP-1, GLP-2 in the case of the pancreas, and then we have actually in the intestine oxymotomodulin, GLP-1 and GLP-2. And I want to give you an example of this, and this is actually work from Merck that is published about trying to get different peptides and different dual peptides, trying to mimic what happened with oxytomodulin. As I told you before, oxytomodulin binds to both receptors. And actually, what they did here is they modified the glutamine versus glutamate to have more specificity to the glucagon, so having the dual peptide versus the GLP-1 specific peptide. And then they add, actually, they add the cholesterol to increase the half-life of these different peptides. And you can see here, actually, in this table, the differences in cyclic MP between the glucagon per se, oxytomodulin, the dual, that is very similar to oxytomodulin, and the real GLP-1 analog. And the effect, actually, in glucogenolysis in the liver. So, they show really that this is a dual and this is mainly GLP-1. So the question was, and is a dual peptide better than a GLP-1 alone peptide? And the other question is, in the case of oxytomodulin, you are actually binding to both ligands, the GLP-1 and glucagon. So mainly you are agonizing the glucagon receptor, and that is good and not that is bad, because as you know, glucagon is not a good thing to touch because you will induce hyperglycemia. So with all this together, if you see the results, actually you can see in body weight mainly a very interesting and nice decrease in body weight with the dual in comparison with the single GLP-1 peptide. And actually, this is not related to food intake, but also there is a change in mainly fat in these animals. And by the other side, they perform an IPGTT, and you see that actually both peptides are very similar with respect to glucose and glucose tolerance. So there is no mainly effect in HbA1c and glucose tolerance, but mainly the big effect is in body weight. And this was mainly surprising because it seems that glucagon doesn't have like a bad effect in glucose tolerance or glucose homeostasis as we expected, but by the other side have a very good effect in body weight. And this is mainly, the effect is mainly in appetite regulation because glucagon is known to do that, but also not only in appetite regulation, probably in energy expenditure. And the second example is, this is from the Markey, this is Walid's friend, and Matthias Schopp, where they synthesize different chimeric peptides in certain amino butyric acid in the second position, and this is to try to avoid the PP4 cleavage, and the lactam to give more glucagon selectivity. And the same question, they have exactly the same question, and you can see here again, for body weight, the lactam peptide is better than the non-lactam peptide that have less glucagon selectivity. But then if you see about blood glucose are almost the same, and this is blood glucose and insulin level. So actually, this was very surprising results because means that actually targeting glucagon at the right way, in the right way, with GLP-1 together, you can have like a nice regulation of whole physiology in the body, and you can actually have a better peptide for body weight in this case. But this is only one example, but you can do exactly the same if you begin combining each of all these peptides that I show you that were in the list that are secreted actually by the intestine. And talking about a little bit about the different modalities, if we compare small molecules, peptides, and antibodies, with the three of them, we can have very good efficacy. The toxicity, we know that we can have great selectivity with peptides and antibodies. We need to be a little bit careful about small molecules because you always can have surprises. The delivery is much better because you can have oral delivery with small molecules, but there are a lot of things going on for peptides and antibodies just now. And you may know more than me about all these different ways that you can target delivery and devices. The regulatory environment is very good, and the manufacturing is achievable. And if you think about the top selling, and this is actually a little bit old, but still is the same thing. There are top selling biological brands over there. The business case is there. You can make a lot of money with biologics. And when you talk about the peptide cells, if you compare, actually from 1999 to 2009, has huge increase in the sales of peptides. And actually, this is growing. This is growing because we will have more peptides in clinical trials and more peptides moving forward. So the peptide market is nearly twice as fast as overall pharmaceuticals, and this is because of the number of peptides as therapeutic targets. And as I told you before, I believe that this will be revolutionary, and this is even growing. Because mainly, if you think about new targets that are coming now in this area, in cardiovascular, in metabolic diseases, not only bariatric surgery, but also in regeneration of the pancreas, regeneration of the heart, are all peptides. So if we talk about what happened in 10 years, this will be very exciting. Then we have improved delivery options, and we have advanced intranasal system. We have buccal inhalation. Many people are working in oral peptides. And actually, this provides really good opportunity for poorly tractable small molecule targets, as I talked to you before with GLP-1s, where you have the GPCRs-B and actually very difficult to tract. And you can have very rapid clinical validation. If you have endogenous peptides, you can go directly into the clinic, infusing them, and trying to find out if they are doing anything or not. So it's a very fast track to move forward. So this creates a double-digit market growth with more than 40 therapeutic peptides in the clinic, and we have like a lot of clinical phases and tons of in the preclinical. So this is very exciting, and why peptides for these areas? I told you before, this is an expanding time. There are new peptides, hormones involving metabolic diseases. I mentioned this before, that they're involving food intake, satiety, energy expenditure, glucose homeostasis, pancreas regeneration, and actually small molecules are very difficult to obtain, and the fast track for all these peptides will be moving as peptides per se, not small molecules. And if you think about the peptides therapeutic databases, there are like a very interesting number of peptides in development today. And also this number of peptides have increased. If you imagine, in the 40s and through now, it has a huge increase of the number of peptides entering clinical studies. And the most interesting thing for us that we're working in metabolic diseases or cardiovascular, if you sum actually the one and the number of peptides in metabolic and the cardiovascular, this is a huge number of peptides that actually are today in comparison what was actually 20 or 10 years ago, that actually only maybe were cardiovascular or more oncology and allergy and immunology. So this metabolic area is really expanding, and we're moving forward with this. So Of course, there are opportunities and there are weaknesses. We can get very readily available leads, and the synthesis and the SAR are very straightforward and rapid. So there is a large number of diverse and natural amino acids available to increase the stability. We can actually access large binding surface areas than small molecules, and this can be a huge problem because actually you want to have very effective way to agonize those receptors. We have the possibility to achieve high potency and efficacy and high selectivity and low toxicity. And for chronic diseases, this is one of the main issues because we really need very, very clean drugs, effective and clean drugs. And the weaknesses, you can have some complex of the synthesis and cost of goods, but for peptides, I don't believe so. My experience with peptides is actually that the main cost will be the syringe and not the peptide per se. Can be cents if you have a good process and production. So I believe that this is not, depends on the peptide that you're talking about. High clearance requires some optimization, conjugation, and formulation that we talk about, and the delivery. We need to think about new ways of delivering this type of peptides. And access to intracellular targets is unlimited, and for this area, actually, I will be a little bit cautious. Still we have a lot of targets that are extracellular or are secreted targets, so I will go for that now. So there is a huge future for peptides and has been a huge interest in the last decade. There is an increase in peptide size and complexity, increased numbers of new candidates, as I showed you before. There is an increased diversity of therapeutic areas, mainly metabolic and cardiovascular, leading the number of peptides coming across in the preclinical and clinical studies. And then there is an increase in the number of marketed peptide therapeutics. And then, if you analyze the probabilities of approval, actually is twofold higher than any small molecule. So I think that there is a huge opportunity here to move forward with peptides. So in conclusion, I want to convince you, I hope that I convince you that there is a huge unmet medical need here to have new safe drugs that are modifier drugs. And I personally believe that the only way to do that is to combine 2, 3, or 4 different entities in one molecule, give to the patient, and as Andrew said before, try to actually mimic what happened in nature. And I believe that that will be the only way that we can stop or actually reverse the disease. And for that, I personally believe that the future is in this area. And mainly I'm very excited about peptides. And I want to thank actually all my collaborators, not only MedImmune, but also my old colleagues in Roche that actually helped me enter in this, I believe, new way of doing things, and in the peptide world, especially Walid that is here. And thank you so much.
Thank you very much, Christina. Do we have questions? Walid, I can sense that you're going to come up with a question. Come on.
No. I want to, the slide number 8 from you about this beta cell preservation. We discussed it long time again from Roche and everything, and we came to the conclusion there it's hardly to go to a normally healthy man like anybody else and tell him in 20 years your beta cell will decrease, so you have to take drug from now. And not only to convince him he's healthy, that he will be sick in 20 years, but who is going to pay for that? And therefore, it's really very difficult to have the insurance company pay for this type of drug. Even if you come with a drug which will preserve your beta cell in years to come, nobody will pay for it. How do you convince the people to first to take the drug?
I'm telling you, well, it will be very simple. When you get actually diabetes, we are diagnosed actually over the years, and I show you very well. you begin having all these complications that actually are extremely costly, especially diabetic nephropathy. If you can avoid diabetes nephropathy, cardiovascular disease with a good drug, I believe that will be very convincing because actually you will save a lot of money from the society, a lot.
The insurance will pay for it?
Yes.
That's what you say.
Christina, one question. You showed some really nice data about the effect of the bariatric surgery on the increase in some of these peptides. Is there any quantitation done and the variability as far as the different patients and how much in one patient it goes up versus another one? Is it dramatic order of magnitude difference?
Yes, well, that is extremely interesting. Actually, there is data, for example, for some of the peptides, it's already known that, for example, obese, morbid patients have a decrease, a decrease in GLP-1, sometimes a decrease in PYY. And actually, there is a variety of increases, I will say, depends on the patient. Interestingly, for example, I'll give you one example. I think that, the data that I saw for some, I will say experiments, but these are a few patients, so depending on the patient, you have the increase. Basal, you will see 2-3 folds increase only, but then the big thing is when you have an OGTT or after a meal, that actually the increase is huge. With respect, for example, there is one study showing about body weight, only talking about body weight. Yeah, there are some patients that actually, after bariatric surgery, you need to wait 3-6 months to see some body weight reduction, but there are some patients that actually they don't reduce any weight, none. So are really resistant to body weight reduction. And actually, they look at those patients, they measure some of these incretins, and actually what they found is that in those patients, PYY was not increased. That is what you expect. So, this investigator says maybe PYY is one of these actually peptides that because of in these patients is not increased enough, you don't have any body weight reduction at the end. So it's a lot of work going on today to really understand how much you will need and how much actually, will get you resistant to this. But the interesting thing is that when we develop this type of peptides, the good thing is that actually you can have some personal health care approach. Because actually you can measure these peptides in the blood of these patients and actually see how they are and how much you will need to contrarrest that, or actually choose the best population that will be more effective to have. Those populations maybe that have the lower levels of these peptides are they going to be the more, the more responsive population. But this is a very interesting point. But just now, there is a lot of work going on to understand more about the super responders and about the ones that don't respond at all, to see how much, which of these peptides is important and actually how much of it. And then, what we cannot forget is that there are a lot of new peptides that are not even in the list that are coming along. So actually, it's a new discovery, very, very early preclinical, where everyone is trying to find out novel peptides that actually are secreted by the intestine after bariatric surgery that are not in the list. So it's a huge explosion of new peptides coming in the field, I will say.
I think that your comments there at the end about the huge preclinical and early discovery excitement is my type of a question here, because I want to shift gears and say, from somebody and as a VP of research, how do you resource this area so that you have the right skill sets of chemists and that hiring that resource? Is the business behind in terms of its realization of who they need to have in place to do that kind of work?
Well, depends on the company that you're talking about, but I believe that if you really want, this is my opinion, okay? I believe that for this type of area like diabetes, you have already drugs there that will reduce glucose, okay? So I believe that what you would like to have is something that goes beyond glucose that actually will be very effective and will try to stop or cure in a way. So actually, if you think about that and actually you convince everyone that this will be the drug that actually will do it, I believe that you will have the resources to combine not only biologists, I will say, but have to be like a good group of peptide chemists working together to try to actually isolate these peptides and actually do the biology on all these peptides and synthesizing and whatever. And actually, there are some people that actually are doing that. Not maybe as fast as we want, but I believe that if you believe on it, you need to do it. And I believe that you will have news very soon about what is going on, in this area and the new type of things that are coming along.
Thank you very much,
I don't know if I
Christina.
answered your question, but
Please join me in thanking Christina.